Okay, hi, good morning, everyone. Thank you for your time to join us for the guest seminar, and thank you for Dr. He for your time. And I will give a brief introduction of Dr. He, and then we can start it. So Dr. He is an assistant professor in mechanical engineering at Lamar University. He earned his PhD from Clemson University in 2011, and Dr. He's main research interest is on computational multiphysics fluid flows, and he has been developed some novel numerical method to solve some complex fluids problem, such as bioprinting and supercritical fluids and nanofluid. And Dr. He will be talking about fluid dynamics analysis of inject-based bioprinting process today. So again, thank you for Dr. He, and the floor is yours.
Okay, great. Thank you, Kevin. Thank you, Dr. Xu, for this opportunity to present my work here. Let me share my screen. Okay, so okay, so so today my talk is on the fluid dynamics. Oh, some technical issues. Okay, all right, we get back. So our talk is on the fluid dynamics analysis of inkjet based bioprinting process. So I'm very happy to be here and to share research with everyone. So I know UDC is a very beautiful campus. I see the picture there. So this, I heard of the program from Kevin, Dr. Xu about NASA CamStar. It's a very nice program here, and I'm very honored to be here to talk and share my research with everyone. And today, first I'd like to give a very brief background for myself and my research interest, and also our HPC high performance computing facility, some brief introduction, and my focus is on research background of this bioprinting work. And my approach is mainly from numerical methods. So then I will, second part, I will do the introduction and some analysis of how we did the work. And then eventually we see the results and we see the conclusions. So basically those the main structure of today's talk. And so briefly, yeah, I graduated with my bachelor degree in Fudan University. I studied my PhD in Clemson University, the Tigers. Yeah, those afterwards, yeah, I decided to find a job in my home country in China. So I was a faculty there in Southeast University for 2 years. Then I went back to the States, do my postdoc and research scientist work in MIT mechanical engineering. In 2016, I get my assistant professor position in Lamar University. So talking about Lamar University, so it is a public university in Texas. It's a part of the Texas State University system. So we have about 15,000 students, and we offer more than 100 degree programs. And if you see our location, right? Our location is east of Houston, but we are on the border with Louisiana. So in the past few years, we have lots of hurricanes coming visit us. Oh no, that's really, yeah, quite harsh, quite difficult in previous summer. Every summer we were very kind of much tense about. So see the beautiful campus, but the campus was damaged a lot during several of the past hurricanes. So surrounding us, we have lots of petrochemical industries. So we have ExxonMobil, we have Motiva, and we have Chevrolet, and many surrounding us. So it's a very active chemical industry there. And so for me personally, I have three different areas. And so wetting and fluid structure interactions, the first part, is also the part I like to share today for bioprinting study. And then I also work on lots of complex fluids. So those complex fluids, different from the previous fluids, is we have a multi-component, like different substances, like we could have different like CO2, supercritical CO2, we have hydrocarbons, we have different other substances all together. So with the complexity of phase change, it's pretty hard, pretty complicated in the composition. So because of that, we have very complicated mathematics representation. Modeling would be quite complex. And another part is recently I started to work in the past few years. I work on the augmented reality and virtual reality research, educational research. So we try to incorporate AR and VR into classroom teaching. So that's part of the work I've done in the past few years. I have been keeping working on. So we on campus, we have two HPC clusters, high performance computing clusters. So those are not so very big ones, as you see here. Right now, due to the 1/4 unit blade system, as you see here, we have very compact but medium-sized cluster. So in the old times, this size of the cluster would take several racks, but due to this blade system, we have compact blades, so we actually pack everything in one rack. So this is a medium-sized cluster. We have or maybe nowadays we should call it a small size cluster. 44 nodes. We have in total 1,564 CPU cores, and we have very good CPU, but moreover, we have a high speed interconnections. We have 100 gigabit per second interconnection, which is critical for lots of for our calculations. Most of our parallel computations fully rely on the high interconnection. So it's not internet. We know internet speed currently is not that fast. Well, yeah, but this is considered to be super fast. So we also have another HPC cluster on campus. We also use lots of HPC facility. It's Texas Advanced Computing Center at UT Austin. So we use them as well. And um, now let's talk bioprinting. So bioprinting is, well, I think 20 years ago is a fancy story, like, oh, we can print tissues, we can print cells to form tissues, form organs, as you see here. The roadmap, this roadmap shows that, well, if we can print an individual cell, right? So eventually we can let the cell to form a sheet of, well, it's a tissue, like a skin-like tissue. And then what if now we print a 3D, a cube? Okay, this cube will eventually, because the cell will grow, if we keep the cell to be alive. So the live cell will eventually fuse into a structure. So from skin-like tissue to a block structure. And what if we form complicated structures like, hey, a blood vessel? Okay, that's something we can do. So if we can do a simple blood structure, a vessel, how about we have the bifurcated vessel, like we have those two branches, two smaller branches in the hierarchical structure, right? So multiple hierarchies are formed together, right? Eventually, you see what if we plot multiple cells in multiple geometries? We have different cell types and we have different structures. Eventually, will it form a more complicated life-like structure? So that's the second stage. And then eventually you see, what if we plot, we print, we design and print very complicated structures and even more number of cells, type of cells, and eventually they will form, could it form 3D organ? Well, nowadays it is very close, or maybe in some areas it is already a reality. So ultimate goal, man-made organs. We may hear some news and we may know some of them are being fulfilled, but still this is not a common practice yet. So we have more and more examples, but these are not a common practice in medical practice. So what are the insights? So the insights here uh, clearly relates to accurate positioning. So because the high resolution for printing is achieved in the technology, so then we can do this type of printing. And because you see we have 2D, 3D design for the blueprints, so then it's possible. And 3D design nowadays is common, it's pretty common. And we have regular 3D printing from plastics to metal. It's just biomaterials. It's not that easy. So we also have high throughput, but well, high throughput certainly it is still a challenge because well, if you are familiar with regular plastics 3D printing nowadays, a desktop printer for plastic 3D printer is about cost about $1,000 to several $1,000. It's in the range. It's achievable for most people, but still, if you are familiar with the 3D printing, you would know that well, printing speed is a little bit low. And sometimes we need to wait overnight or maybe wait for a few days. So high throughput is a problem, especially if we consider we do bioprinting. If the high throughput is an issue, so we may have cell issues, cell viability. Will the cell can sustain those long time for printing, right? So overall, considering all of those insights, the major grand challenge that we have is cell viability, especially for delicate cells. Will those delicate cells survive in the harsh printing process? It becomes an issue. So uh, in order to solve it, we like to say, hey, can we model? Okay, so those are some examples as we see here. So Dr. Anthony Attala, well, he presented a very interesting TED show a few years ago. If you are interested, feel free to find Dr. Attala's TED show to see how he presented a 3D printed kidney. So the thing he's holding is a 3D printed kidney. Okay, so this is a work have been done in his lab, and well, I was collaborating with one of Dr. Atana's former student before. So this is his image. So if you see here, right? Well, I should say that this kidney isn't printed from this printer, okay? But if you see this printer, this is a regular. Um, desktop inkjet printer. So you see here, everything here, you see, we even see paper feeder. Those are paper feeder. Those are regular ink cartridges. As you see here, we have two cartridges right here. And everything is the same. This is a converted from. You even see a paper fed and moved through this platform. So what they did, they just convert a desktop inkjet printer into a bioprinter. And they certainly need to cleanse the cartridges, and they need to make sure the cartridges are fully converted to be bioconvertible. So then they put cell solutions, they call bio ink, into their printer cartridges, and then they convert this platform. So they have a vertically controlled platform. So, well, of course, in our inkjet printers, we don't need, because we just print 2D images on paper, we don't need to tune this platform, but they did. So they did create a vertically controlled platform. They can move up and down, move up and down. So, and then they have used hydrogels, so cross-linked hydrogels. So what does it do? So they have hydrogels, and they have cell solution in the calcium chloride liquids. So cells represented in red. And when the cell injected into these uncross-linked hydrogels, and calcium chloride will trigger the cross-linking reactions for the hydrogel. So this uncross-linked hydrogel is kind of a oil solution. So it's very sticky, viscous. In fluid mechanics, we call the fluids as viscous fluids, highly viscous fluids. So calcium chloride will react with the hydrogel, and the hydrogel will have cross-linking reaction. Cross-linking will fuse the structure together, so the cells will be fused together. Okay, so this is the physical configuration, and we also see examples from actual practice, right? So how can we solve it? Well, the reason we want to solve it is we want to understand how the cell, uh, experienced the harsh printing process. So then we need to decompose the system to see, hey, what are the physical models we can use? So physically, how we see that a cell-centered droplet is the one we like to study. Well, in reality, we have multiple cells occupying one droplet. But in order to reduce the complexity, in my former study, I only say, hey, we start with one cell. Okay, one cell. How about we put the cell in the center? The reason we put the cell in the center because we want to reduce the modeling from 3D, three-dimensional modeling to two-dimensional axis symmetric condition. So in order to to reduce the computational hours, computation is quite intensive. It could take several weeks to get the results. So when we reduce the dimension to 2D axis symmetry, we put the cell in the center. So we reduce our calculation time to about 24 hours. And okay, droplet is here, droplet is impacting to the system. And we have this polymeric solution, which is the highly viscous uncross-linked um, uh, hydrogel. So then we have a multi-phase flow, of course. We have the three fluids, and then we have surface tension for droplet air and polymeric solution with air, but we neglect the surface tension between droplet and pool. So when droplet contacts with pool, so we know that they are water-based solutions. So the contact will have no surface tension. But then The second part will be a little bit complicated. We have a fluid structure interaction. So cell is very complicated. So we have different internal structures for cell. For example, the cytoskeleton, and we have the plasma inside the cell. We have different small structures. We have the nucleus of the cell. And so how do we modify cell? How do we model cell? So and how can we translate the cell viability into mechanical properties? See, my background is in mechanical engineering. So how can we translate, how can we interpret the cell viability with its mechanical properties? So this is one thing, right? So next, in the analysis, second part in the analysis, so we see the domain, right? The domain, the physical model. Second part is we do have mass transfer. So remember we talked about the previous one. We have calcium chloride. We have water solution, calcium chloride, water-based solution. And then we have water-based, that's why it's called hydrogel, right? Water-based cross-linked hydrogel. So we have mass transfer. So the time scale analysis tells us that the impact process takes about 100 microseconds, while the mass transfer, the species diffusion, takes 50 milliseconds. So you can see that they are largely different. Because they are largely different, we can assume that the mass transfer and reaction of cross-linking are neglected during this process because we only model very small time scale for the impact. So that's why the cell viability is determined in this 100 microsecond harsh time. So we are going to just see this harsh time. Well, will the mass transfer affect cell viability? It may affect, but we will just focus. So this study, we limit our study to we try to see whether cell can sustain during this 3D printing process. So we focus on this 100 microsecond. So numerical methods in this study is relatively complex, so I'm not going to dig all the details into the process. Okay, so briefly, very brief. So we have fluid-fluid interaction. We have fluid structure interaction. So when we say fluid, so we have different fluids, right? We have the droplet fluid. We have the highly viscous polymeric solution. So those are different fluids. And when we say fluid structure interaction, we have the cell as a solid structure. Well, it has a type of fluid property, but we assume it can be treated as a pure solid based on experiments. So experiments revealed that during the harsh impact, the cell's main mechanical behavior is an elastic body. So because of that, we treat cell as a simple solid elastic body. And then for fluid-fluid interaction, we track the interface using level set method, and we treat the interaction force, use the two methods. One is called continuum surface force. One is called a ghost fluid method. So the two are alternative. So sometimes we use CSF, sometimes we use GFM. We try to compare them. Eventually, based on experimental data, we concluded that CSF is slightly more accurate, but they are very similar. So in the fluid structure interaction, we also compare two methods. So one is in the literature, we have a ZFM method. One is the one I developed in this research. It's called a fully Eulerian FSI method. So we are going to see them. And handling interaction, we use the same, actually very similar to CSF. It's called immersed boundary method for fluid structure interaction, but it's very similar to the fluid-fluid interaction, which is a continuum surface force method. So those are the brief introduction of this numerical methods. But because I developed this new called a fully Eulerian fluid structure interaction method, so I like to talk a little bit about this method. Okay. So why this is a fully Eulerian method? Because we track everything using the Eulerian representation. So in fluid mechanics, we treat everything. So say if we use a fixed fixed spatial points to describe material, we call that as Eulerian. If we describe material using the material points, well, the material points will move. So that is called Lagrangian. So in solid mechanics, whenever we deal with elastic or plastic bodies, the natural way is to describe it using Lagrangian description. However, this method, because we are dealing with fluids mixed with solid structures, so a fully Eulerian approach might be better. So then I developed this method. So we can see we have surface interface. So this is a description for the surface interface. It's actually distance. So how far away from the interface to from the location to the interface. And then we also have material coordinates. In this case, we have X and Y. So those are very simple. You can see that. And using this very simple description, right? We can actually model the transformation of the shape. So the shape is transformed from a regular sphere. This is an illustration. So those circles represents fixed points. So that means all the data are stored on the circles. So circle will have the data points. So based on the circle, we describe X and Y. So X, capital X represents the material coordinate X, and Y represents the material coordinate in Y direction. Well, if you see lowercase x and lowercase y, they mean the spatial location. So spatial location means, space, right? We fix space. Unless we are talking about Einstein's general relativity, right? In Einstein's general relativity, space will deform. Space will have its own deformation. However, if we are in the Minkowski space, which means space and time doesn't change. So space is fixed. We have the fixed space, lowercase x and y. But the material is changing. Well, for example, this spherical body, let's say a cell, changes from its location. As you see here, we have translation, we have rotation, and we have deformation. So not only we see translation, we have rotation, we have deformation, as you see here, and all the material deformation and translation and rotation can be described by the coordinate, material coordinate function x, y, uppercase x, uppercase y. So how do we calculate its transformation? Its transformation, we use the matrix d x over d lowercase x, dx over d lowercase y, and d uppercase y over d lowercase y, and d uppercase y over dy. So this matrix, well, this is in 2D. Of course, we can have 3D transformation matrix, and the stress can be computed using this relation. This is called the Neo-Hookean law. So using the Neo-Hookean law, this shear modulus times the deformation. This is the deformation difference. So A times A transpose matrix minus the unit matrix I. So then we can get the elastic stress. So based on the elastic stress, we can calculate the body force, internal body force, B, and we can calculate the solid fluid interaction force, which is tau dot the normal direction of the interface and the smearing delta function of the interface. And of course, divided by rho. Rho is the density of the solution. So essentially, using this method, we can describe any deformation. Like this is one of the moment for the impact. So when the cell, biological cell, living cell impacting to the solution, we see the deformation. This is from our calculation. So previously we see the illustration. So now we see one instant of our calculation. So when we calculate this cell, the strong impact will cause deformation, and it will change the cell. As we see here, material coordinates deforms, and of course they translate. We don't see large rotation because we model this perfect symmetric situation. That's why we don't see rotation. However, in reality, any small imbalance in the actual direction will cause rotation. And the model, our model, can capture the rotation, just that in this specific calculation, we have axosymmetric situation. So that's why we don't see rotation. Okay, and this is the stress tensor component, sigma xx. So we have xx component of this tensor. All right. So now, well, as a numerical researcher, my role is to model it, but my role is also to validate. So I cannot use a model say, hey, you know, this is a calculation, this is a result, and you need to trust my result. I have to do serious modeling validation in order to know, you know, I developed the code using mixed C and Fortran codes. So the codes have been developed. I wrote them. I debugged the code. It took lots of efforts, but you know, in the end, it might be wrong. I cannot guarantee this is correct. Well, even we use commercial softwares, we cannot guarantee the simulation will give us the correct answer. So no guarantee. So what we should do, we need to validate. So my first validation is for pure multi-phase flow. As you see, it has two components, multi-phase and fluid structure. So we can use literature data to verify. So what I did before, I did an axisymmetric flow for this droplet impact. These are my simulation results. As you see here, we have a droplet impacting to the pole, have a velocity, and we have gravity. The velocity is 1.55 millisecond based on literature data. So I follow the literature data. And I use two models. One is called a ghost fluid method. One is called a continuum surface force model. So I compare with experimental data, which represented by the dots. And we can see that surface continuum surface force gives a better representation. So then because of that, I decided to say, hey, you know, after the validation, I like to use a CSF model in my study. Okay, so these are, well, in this interesting video, so I computed this and compile my results into a video. You actually see reflection, so it's very interesting. So then when the wave goes to the boundary, we have a reflection coming back. Okay, so that's really something very interesting because in reality, we have quite large fluid bath. We don't have those quick reflection, but in numerical simulation, we have those reflection. Luckily, our validation just stop here, so we actually don't need to worry about the reflection coming back. And yes, that reflection will actually modify the behavior for the center droplet. So then that means our simulation will be affected if we want to see long time. And luckily, our simulation only focus on small time. It's around 100 microseconds during the simulation, so we are fine. So then certainly we are fine then. Okay, so what about the fluid structure interaction? Remember, we have a cell, which is elastic body during this process. So, and I developed this Eulerian method. So we use level set, solid level set, as you see here. So again, I compare with literature data. Literature data shown here as the dots, and you can see the lines are my results. So we have a cavity. The cavity moves in this direction. The lid moves in one direction towards the right. And because the lid is moving, the fluid flow drives the vortices inside the cavity, and that vortex of the fluid's motion will deform the solid, which is an elastic solid on the bottom of the cavity. So that's why you see here vortices, right? So this is a mix of liquid and solid. So I didn't show the liquid part, but if I show the liquid part, you will see the four vortices is actually a bunch of vortices going in one direction, do the rotation, okay? So I only show the solid part, but the solid part has been affected by the fluid motion in the top. So we use the we use the field extension so that we make sure near the solid boundary, the solid coordinates, the material coordinates, are not affected by the fluid motion. So that's why you see the mixed reality. Fluid and solid are coexisting in the same domain, but it's just numerical issues on the top. So we have good agreement with literature data. So after the validation, right, we validate, as you see here, we validate with a shear model, shear modulus for the solid, viscosity for the fluid. We model everything the same as the previous literature study. So now we are sure that we have a good implementation of both parts, fluid multi-phase flow and fluid structure interaction. So after we have both working, so now it's say, hey, how about we put the cell in the bioprinting process? We want to say, hey, what's going on there? Okay. So here is a printing process as you see here. What if we only print a cell into water? There's no polymeric solution, just a cell into the water bath. And as you see here, cell represents the represented by the green ball. The ball is pushing down, and I should note that there's no initial velocity. So the process generates a very high speed. We have almost 1 meter per second for the droplet motion. So initially it's a zero, it's a stationary station. Um, Stationary situation, basically zero velocity, but we generate actually 1.5 meter per second velocity, which means a harsh process for the cell. Where does the energy come from? The momentum, the generated kinetic energy is converted from the surface energy from the initial conditions. So we say, hey, that's why water is not good for the process. We don't want to use pure water, and we cannot locate the cell well, and we will cause lots of a harsh process. So, okay, that's the case. So then we say, hey, the representative case should be exactly the same as the actual experiments. So then we do the this testing. So we have a droplet, 60 micron. It's a 1 cp. Its viscosity is 1 cp. It's the same as water. And the cell is 30 micron. Well, it's a typical size for a cell, for the cell. And then the cell modulus, shear modulus, is a 10 kilopascal. It's regular. And bath is large, right? The best size and the best viscosity is pretty large. It's a 20 cp, so 20 times of water is kind of a high viscosity we have. The initial impact speed is 4.5 meter second, so it represents inkjet printing. So the reason inkjet printing is favored in this research is because of high throughput. So in order to have high resolution and also high printing speed, so inkjet is essential, is better um, the um, than the slow speed methods, like if we squeeze the cell solution onto the printing base, well, we may not have this harsh process, but on the other hand, we may have very low speed and also accuracy positioning will be low resolution, so that will be also the concern. So essentially, this process only takes about 75 microseconds. Let's take a look. So, well, it's an ordinary case, right? So it pushed down and the cell changes its shape. And you can see that we have this tip, the droplet top surface pushing down and squeezes the cell top surface. Okay, so let's take a look. This is the process, right? Let's take a look at the, if you want to, well, this is the benefit of using numerical simulation. So if we model and simulate this process, we can actually get the data from almost every part of the simulation. We can abstract the data, we can put them aside, and we can analyze, for example, cell deformation. So the color distribution represents the von Mises stress. Well, this is the idea in mechanical engineering. In solid mechanics, we use von Mises stress to see the stress, whether the stress can be damaged. So usually we try to see whether certain material is damaged due to high von Mises stress. So the calculation takes like this. So sigma, we have sigma at different, so stress at different direction, and we add them together, square law, and add them, divided by 2, we have square root, give us von Mises stress. Okay, so we see that initially deformation takes on bottom. So we have bottom deformation due to impact. And essentially the deformation changes to the side on the top. And then the top is pressed down by the fluid flow, and the top forms a large deformation and stress. Von Mises stress increases. And the largest stress appears on these flat surface corner. Well, that's because the shape is changed a lot. And then after it's coming back, it's recover its own shape, the stress reduces essentially to zero stress. So this is the cell deformation process, right? So in order to know, well, we identify this is the harsh process, very large stress occurs right here. And how can we know which is the best way to do cell printing to help the cell survive? We did parametric study. The parametric study tell us, well, we study different impact velocity, we study different pull depth, we study cell modulus, we study pull viscosity, we study drop viscosity and surface tension, right? We see those different behaviors. So we actually identify sigma, surface tension, is the most important factor for cell deformation. So based on this detailed study, well, I'm not going to show all the details a lot. We did more than 100 simulations to identify different conditions. Based on the 100 simulations, we actually find a general picture for bioprinting. inkjet bioprinting. We identify that we have inertial stage in the beginning. The time is around U over R. R is the radius of the droplet. U is the initial impact velocity. And afterwards, we have inertial interfacial flow stage 1 and stage 2. So during the stage 1, it reaches the maximum stress. And this is a stress, vertical is a stress. And the time and interfacial flow stage 2 is going down, right? So in total, we have the time according to the surface tension, sigma. And afterwards, we have elastic response stage. And in this response stage, because of the elastic nature of the cell, mainly contributed from the cytoskeleton structure inside of the cell, and the cell stress will increase, but it's a small peak. And essentially, it will go down, and that is a viscous flow stage. And the total time is governed by the viscous flow stage. So the maximum stress is around this is on the order of this magnitude, sigma surface tension and viscosity and the diameter of the cell. So these are the basic general picture. How can we promote cell viability? Make sure. So one way is the way I propose. Can we use electrohydrodynamics? Well, EHD is called electrohydrodynamics. So we use electric force. Well, electric force has two parts. One is induced by the charges. One is because of the difference of electric permeability. Air and liquid water has different electric permeability, so that will cause electric force. So again, we neglect the cross-linking, we neglect the mass transfer, but we will see, hey, can we use electric field to change the behavior? So this is what I did. I model here, we have electric field. This electrode is on the top, and top electrode and the bottom is the liquid is the bottom, the yellow surface. So this is a cut view. And we do see that if we have a 200 kilovolt electrode here, so it does change the liquid behavior, but it doesn't change too much. You can see that, right? So it means, well, maybe we should manipulate, we should change the electrode, right? Indeed. So if we enlarge electrode on the top, if we actually reduce it to 100 kilovolts, just to reduce the voltage, but enlarge the electrode. This is one thing. Second, we tune the dynamic control. We have a dynamic control of the electric field, as you see here, initially very large, but gradually we reduce essentially zero. So we do see that the top isn't pushed that harsh, as you see here. The top isn't pushed that harsh, but we also induce some liquid flows. That's okay. That's certainly okay. We have some liquid flows. That's fine. So after the calculation, we see that without electric hydrodynamic force, so we have very harsh process, but with this controlled electric hydrodynamics field, we can reduce the cell stress, formicis stress. But we do have the elastic response. You see here, the second peak is electric response. So that means we need to dynamically control. So this is not the best control scenario, but we successfully reduce the cell stress. Okay, so we also see this EHD jet. This is typically a cone structure, is well reported in the literature about the electric hydrodynamic flow. So we do see them. That's okay. That doesn't affect the cell. So let me give the concluding remarks. So majority of the work I presented today is done in my in my PhD stage, and the EHD study is done in my current work. So during this work, we see that a bioprinting process can be modeled as the cell-laden droplet impact onto a viscous liquid pool. Cell deformation and cell history is important. We study them. And different printing conditions have been explored. And we see that different stages with different emphasis of which one is more important. We know that this will help us to design new technology to improve cell surviving rates. And certainly one thing I did is using EHD to help improve cell surviving rate. And we know that, well tuning, so signals will be important. So certainly there will be continuing work to optimize which is the best tuning signal for electric fields so that we use to help cell to survive. Okay, so I have some acknowledgement for my funding and for my computational funding as well. So I have my doctoral student graduated in 2020. He has been working in Penn State as postdoc for more than 1 year now. These are my current students, and I have lots of undergraduate students working in my lab. And I also have collaborators from different places in the States. And so with all, I will be very happy to answer any of your questions. I appreciate your time and your attention. Thank you very much.
Yeah, thank you very much, Dr. He. Yeah, this is very impressive work you shared with us today. I just have a kind of a, I think your research, is very relevant to, to some of the work. We were primarily focusing on experimental studies here.
That's great.
But very impressive high performance computation, facility, and also, the models you have developed. So one thing, similar to what you have worked on, we are looking at some either it's called aerosol jet printing, or
Nice.
one of the system we're trying to get into the lab is called a nanojet printing. It's very similar in terms of the principles. So of course we're focusing more on the experimental side, but I will be very interested to know whether we can work with you to see whether we can leverage some of the models you have developed and the expertise in the modeling to see how those process can be understood using your computational methods.
That's great.
Yeah. And the other thing, my student is working on something a little different scale, but it's more for the thermal elastic resins. Because we work with NASA, so there's one of the materials they're interested in for building the thermal shield materials. So maybe he will ask you some questions later, but one thing at least came to my mind while listening to your talk is we probably can see how your model can be used to understand because it's going to be a mixture of resin with some other materials, so it could be useful to kind of have some simulation model to understand how different mixture can change the viscosity, how the flow will be able to be impacted.
That's
so very interesting work. Very interesting work. I see a lot of the application from your work to what we are doing here.
Thank you. That's good.
I don't know so I actually have some students in my class here as well, so I don't know Any of them have any questions they want to ask?
Yes, good afternoon. Can you hear me?
Yes, we can.
Hey, good afternoon. Thank you for sharing. That was fascinating to follow. I'm actually the additive manufacturing student here that Dr. Zhu was referring to. This is my world that I live in, so it was fascinating to follow. I'm actually from going to and from right down the road from you at Lamar. I was raised in Baytown, which is just to the east of Houston.
Oh, close to us.
Yes, sir. I have just two, one very just for my own interest question, and then one serious question about your research. On your high capacity computing, how much do you have to focus on cooling and keeping those types of processors cold while you're running them at that high level?
Yes. Well, yeah, that's a very very hot topic in most high performance computing centers, and I know Google did a lot in this their cooling facilities, so they proposed and developed their own in-house cooling solutions. Well, for us, we are quite lucky. So as faculty, we do not need to do anything about cooling for or improving the current data center work. So we have our own IT department. So their focus is to provide a computational facility to us. So for me, I'm the director for the current HPC facility on campus, but I only concern about the hardware of my cluster and also the software configuration of my cluster. So they actually do everything. They set up the data center. We actually have very advanced cooling devices. So almost every stack, so every two stack share one cooling unit. So I was quite amazed, they did a very good job there.
Wow, excellent. Yeah, I know that's one of the key components to how high you can run those type of computers. So I imagine that's a very important focus for them.
Definitely.
Second question I had is I didn't see, and maybe I missed it, have any of those organs been used, or are they viable to be used yet, or is that just getting to the shape and general size of the organ right now?
Oh yeah, that's a very good question. So you see, in my background, so my focus has not reached to the stage, but when the cell is formed, or when the tissue or the organ is formed, and how would they make the use in actual transplants? Well, they did it in mouse many years ago, and I guess nowadays human test is already past the government licensing issues, so they already did lots of work. But that you are right. So it's just the reason you didn't see is just limited by my background, by my research, activities. So I haven't touched that area yet. But certainly, it's important. After the printing, the researchers, the actual experimentalists, need to make sure most cell will survive, and they will be cultured so that essentially they're going to transplant the structure into a living animal, and then they are going to observe how will that printed structure be merged with the host body. So that's actually very interesting research. It's just I didn't do those work.
Right, I understand. Thank you very much for your presentation today.
Sure, thank you, James.